Okay, we're going to continue on with patterns of inheritance here, but now we're going to talk about some of the more advanced things that Mendel didn't address directly. Remember, we talked about Mendel as um, solving kind of a crisis in biology, dealing with inheritance. Inheritance initially seemed too complex to understand. There were hidden traits that could disappear and reappear at seemingly random intervals. It was also hard to figure out how one cell could carry all the information to make fluids, so it was thought that maybe it was in the fluids that were male fluids and female fluids, and so there was this blending idea. Um, but Mendel came from an agricultural background, and they had been working on this problem through selective breeding for actually hundreds of years, and were applying principles of science, recording the traits of the offspring and controlling the breeding of both plants and animals. A good example of this is how effective they were on merino sheep. The sheep you see in the upper photo is the same as in the lower photo. It's just in the upper photo, the merino sheep had been separated from the flock for a year and not shorn of its wool. Selective breeding has so increased wool production that this animal is actually dependent on the farmer now to remove the wool because it actually can make him like a fur ball. And this was from hundreds of years of farmers breeding sheep for wool. They would breed the individuals that produced the most wool, the best wool, and if they had an individual that was particularly good at wool production, even if the offspring weren't that good, they would continue breeding them to relatives until they got the traits that they wanted. And so Mendel was aware of selective breeding, how effective it was. And so he used agricultural plants that he knew well and could control the fertilization of. And he followed specific traits that he knew were particulate. And he bred true breeding plants in this test cross and then recorded the numbers of offspring of each type and then worked not only on the first generation but on the on the second generation. And this helped explain why selective breeding worked. That traits were particulate. Individuals had two alleles for each trait. Alleles could be dominant or recessive, and individuals would pass on only one allele to their offspring, so they would segregate during the formation of gametes. Inheritance for one trait didn't affect the inheritance of other traits for the traits he chose, but this isn't necessarily always true, and so we're going to see that today. And so here's kind of the Punnett squares for Mendel. He looked at one trait, flower color, and identified purple as dominant and white as recessive. When he bred two breeding parents in a test cross, all the individuals were purple. Okay. When he allowed those individuals to self-fertilize, you see that in the lower Punnett square, he got a ratio of 3-to-1, a phenotypic ratio of 3-to-1, and then he figured out that the genotypic ratio was 1-2-to-1, so that there were heterozygotes. And this worked for all 7 traits that he followed. But this didn't explain everything. Okay, it didn't explain certain traits that had continuous variation. Traits like height, weight, intelligence. Here the variation isn't discrete, it's continuous. Most individuals have intermediate forms, but there are individuals with a whole range of characteristics. And the explanation for this is that this trait is affected by many genes, by genes that affect food processing, that affect metabolism, that affect activity, that affect production of hormones. And so there are multiple genes that affect this, and so it's going to produce continual variation. There's also strong effects of the environment. For any of these traits, if you give individuals less food, they won't be as tall, they won't be as heavy, they may be less intelligent. The environment affects the expression of many genes. In some cases, we can see that really directly. There's a Siamese cat right there. Coat color in rabbits and some cats is affected by an allele that produces an enzyme that only produces pigment at temperatures below 30 degrees. And so this is only going to produce pigment in cooler areas of the skin, the tail, the ears, the parts of the face, parts of the feet. This is a good example of how environment can affect some genes. Okay, there are other genes that affect more than one trait. Okay, so that several traits may be affected by an allele, and that will affect its inheritance. This is called pleiotropy. Okay, and here we have hemoglobin alleles, or alleles that affect hemoglobin, but also affect resistance to disease. And so in hemoglobin normally carries oxygen. We have a normal hemoglobin allele, which is dominant, which is very good at oxygen transport. Then there's a sickle cell allele, which isn't so good at oxygen transport, but confers resistance to malaria. So homozygous individuals, they are good at oxygen transport, but they're susceptible to malaria. And so they often die of malarial illness, fever, and anemia before they reach the ability to pass on their genes. And individuals that are homozygous for the sickle cell gene are resistant to malaria but have a poor oxygen transport and die of anemia. Here, heterozygous individuals have both the ability to transport oxygen and resistance to malaria. And so this gene is common wherever you see lots of malaria. And it's a different pattern of inheritance, and it produces more than one trait. Likewise, sometimes there are genes that are linked to other traits. Okay, this is also different from what Mendel described. These genes are carried next to or on chromosomes where other genes are carried, and so the traits are linked. They appear to come together. And in this case, sex-linked genes, they're carried on the X chromosome. The X chromosome is a big chromosome. Not only does it carry the genes for determining a female, but it carries genes for other things like blood clotting. And so hemophilia is caused by a defective blood clotting gene. And so in humans, human females have two X chromosomes and have two alleles for the blood clotting protein. And so they can be heterozygotes. And so the normal blood clotting protein is dominant compared to the hemophilia gene. But males never really have heterozygotes. They only carry one allele because males have an X and a Y. The presence of a Y determines whether you're a male or a female, and so males are male. But the X chromosome here only carries the allele for blood clotting. The Y chromosome doesn't. So these individuals are going to express whatever gene they have for blood clotting. And so they always express recessive alleles. So here is a pedigree chart for the British royal family. And Queen Victoria was heterozygote for hemophilia, which meant she had normal blood clotting but could pass on a recessive allele, okay, as could her daughters. Okay, and one of her daughters married into the Russian, or one of her granddaughters married into the Russian royal house and carried this recessive allele. And so this recessive allele was passed on to the heir to the Emperor Nicholas of Russia. That's Alexei, a very sick little boy that had hemophilia. This was a very big problem and so preoccupied the Russian royal family that it in part led to the Russian Revolution in communist Russia, something we're still kind of dealing with today. Another kind of non-Mendelian pattern of inheritance happens with the human ABO blood groups. Here, this system demonstrates that there can be more than two alleles for a trait. There are three alleles, and two of the alleles are codominant, always expressed when they're present. Only one is recessive. That's the little I allele. There's a typo there. It should be a little I. Okay, and so here, individuals only can have two alleles, but instead of having just homozygous dominant, homozygous recessive, and a heterozygous, here you have a bunch of different homozygous. You can have a homozygous recessive, type O, two small i's. You can have homozygous dominant for A, homozygous dominant for B. You can have heterozygous, where they're codominant, so you have AB blood type. And then you can have heterozygous with the I and the IA blood or the IB blood. And so you have all these different types of blood donors and blood types. You can be type A, type B, AB, or O. And if you're type A, you could be dominant A, homozygous for A, or you could be heterozygous for A and O. So it leads to more complex patterns of inheritance. And so this points out that while human traits are controlled, many human traits are controlled by a single gene where you have dominant and recessive inheritance, that many times you need a pedigree analysis to track inheritance patterns in families. And sometimes these may not come to just the simple two allele systems with dominant and recessive. Sometimes it can be something else. But in many cases, what we see is that it does track what Mendel predicted, particularly with many cases of diseases, because we know of sometimes that these are caused by dominant pedigrees, where it just appears in any time the gene is there, you get the disease. And other times you have a bunch of recessive phenotypes. So recessive phenotypes might be something like the albinism, where you need two copies of a recessive gene to see the trait. So in a dominant pedigree, you'll see if the gene is present, you see it every there, so it will never really skip generations. Once it's not there, it's not there. But in recessive pedigrees, you'll see things skip generations. So you'll have carriers. So carriers, here they're shown as half green, half white. They have the normal phenotype, but they can pass on the genes. And so if you have two heterozygote carriers that produce offspring, some of those offspring can have the condition. So like here, we see in generation 3, the condition shows up where neither parent had it before. And then sometimes it can skip a generation. Okay. And sometimes we see traits that require two alleles to give this trait present. And so sometimes we see even more complex patterns where both traits have to be present, both genes have to be present. And so here's a pigment production that requires two separate genes, and those genes have to be present. And so you can get these really complex patterns where to get a trait there, you have to have two copies of a dominant gene. And so here's a pigment production that requires two genes. The genes have to be dominant, and you wind up with this kind of wild pattern of inheritance where it doesn't follow a 3-to-1 ratio. Okay, so just some examples. So finally, this genetics, this had lots of application in plant and animal breeding. This has also had applications in human traits and chromosome mapping and paternity, understanding evolution in humans, and understanding human genetic diseases and diagnosing and treating those.